Okay friends, so welcome to online session, class 12th, subject biology. Today's chapter is molecular basis of inheritance, session 1, and myself G.R. Chopde, PGT biology, JNV Sangli. So we divide this chapter into 5 sessions. Now what we are going to study in the chapter? The search for genetic material. Then DNA, structure of DNA, DNA replication, how the DNA makes its own copies. Then word of RNA, that is different types of RNA and their main functions in our genetic system. Then transcription, that is synthesis of mRNA from the DNA. Then genetic code, that is triplet. What is the meaning of genetic code? Translation, where that information carried by mRNA is converted or translated into protein or amino acid chain. Then how these genes which are expressed are going to regulate, that is regulation of gene expression. Then human genome project. So how many chromosomes, how many genes, what are the total percentage of genes, like that. And finally, DNA fingerprinting. So today we'll discuss about the search for genetic material. Now try to understand the concept of a gene. We know that the gene is a segment of DNA. It is a segment of a DNA which is responsible for the inheritance of characters, or in another word, it is a unit of a inheritance. So in previous session, we have discussed during the principles of inheritance. G.J. Mendel called they are the factors. Now later on it is named as a gene. So every gene will control one characters like tall or height. They are called traits. So what are the modern definitions of this gene? So it is a segment of DNA having the limited number of nucleotide pairs. Yeah, nitrogen bases the pairing in a unique sequence. So that sequence is very important. Among that, there is a cistron. It is a functional unit of DNA, functional, responsible for the synthesis of RNA, or from that RNA, the protein chain. It was according to the Seymour Benger in 1955. Muton. Then what is a muton? Okay, so this is what you are now, a cistron. Now second one is a muton. Again, it is a segment of DNA which undergo mutation, that is sudden change. We have discussed in last chapter also. Then the third category is, that is recon, the shortest segment of chromosome which undergo recombination, and recombination discovered by a Morgan. Okay, along with mutation, we have studied again that in last. Okay, but to remember, these are cistron, muton, recon. So these are nothing but a genes only, but having different functions. Now, so how that search of genetic material begins? It is a Griffith's transforming principle experiment. Yeah, principle experiment. Frederick Griffith in 1928 used a mice, or even rat you can say, and a bacteria Streptococcus pneumoniae, which cause pneumonia disease. So Streptococcus pneumonia has two strains. You can see here. So there are two strains. One is the smooth strain. Okay, that is virulent. Because the smooth strain we call a S strain. So it has a coat, glycoprotein, a polysaccharide coat, mucus coat around that bacteria. So that is responsible for a disease that is cause pneumonia. So therefore it is virulent and poisonous. Then there is one more strain referred as a rough strain, a R strain. So R strain, the word came because there is no this polysaccharide mucus coat. So polysaccharide mucus coat is absent here. So therefore it is non-virulent and no mucus coat and do not cause pneumonia in the mice or any other organism. So these are the two strains what the Griffith observed during his transforming principle. Now see, he took the rough strain. This is the first experiment, which is non-virulent, means without any coat. He injected into a healthy mice, injected, and you see that the mouse will survive. It does not die. So that is non-virulent. But he injected a smooth strain, which are virulent, into the healthy mice or mouse. So it will die due to the pneumonia disease. And from that he concluded smooth strains are virulent. Now what he did in the third experiment, okay, he took this smooth strain and heat killed, उनको गर्म किया. So due to heat, they killed and they lose that property. He don't know what is that property. But when he injected into healthy mice, he found that mouse will survive. It will not die. Earlier it was died because they are smooth strain, but they're living. Now here they are dead. So from this is conclude, when they are living, they cause the disease. When they are dead, they does not cause the disease. Then he took this first experiment and the third experiment and combined. Okay, that is the rough strain and heat killed smooth strain. So mixture both. See here mixture. So what is the result we got? So the mice will survive live, and here also it is lives. But a mixture is injected into the healthy mice, so he got a other result. So the mouse will die. Then he started thinking why it is so happened. So in examination, you can write in this way also. So our strain bacteria injected into a mice, the mice will die. Then R strain bacteria injected into the mice and mice will live because it is a rough strain. Then S strain, which are heat killed, injected into the mice again survive. But combination of S strain, which are heat killed, plus R strain, which are live, when injected into a mice, the mice will die. So in this way, you can represent in your examination. Now, what is the conclusion of this experiment? R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Okay, somehow he doesn't know how it is. So some transforming principle transferred from a heat killed S strain bacteria had enabled and made the R strain bacteria to synthesize the smooth polysaccharide coat. And these R strain bacteria, because of something is transferring from the S strain, they become virulent in a S strain. So in simple word, the R strain bacteria are somehow converted into S strain. So that is transformation. That is transforming principle. So transformation of R strain to S strain is due to a transfer of genetic material. So this is then later on it came to know. However, the biochemical nature of this genetic material, whether it is a DNA or a RNA or protein or lipids or fats or carbohydrate, because so many biochemical compounds are there in bacteria, he doesn't know what it is. He not defined from this experiment. But what he concluded, something is transferring from S strain, that is dead S strain, into the R strain. Therefore, R strain bacteria become S strain due to the secretion of polysaccharide coat. And that makes the bacteria virulent, and therefore the mice will die. So then there are so many other. See, he concluded that some transforming principle transferred from a heat killed S strain to R strain. It enabled the R strain to synthesize the smooth polysaccharide coat and become virulent. This is due to the transfer of genetic material. So in this way also you can explain. Now, what is that biochemical here characterization? Three scientists, Oswald Avery, Colin MacLeod, Maclean McCurdy in 1933 to 44. These three scientists, you can see the scientists. So they purified the biomolecules from these bacteria like proteins, DNA and RNA along with carbohydrates and fat also. But we will give more importance to these three elements. From the heat killed S cells to see which one could transform in a living R cell to make them S cell. So again they perform the fourth step experiment conducted by Griffith, but by addition of certain enzymes. So here you can see enzyme protease. Heat killed S strain. So there is a protease enzyme. So protease will help to digest the proteins, but not a DNA or not a RNA. Only proteins are digested. So what happened? Plus live strain are bacteria. So transformation takes place. Either transformation हुआ. Heat killed S strain, then they added RNAs, which will digest RNA, not DNA or not protein, along with live R strain. Again, here also they observed a transformation. But in third experiment, heat killed S strain along with this enzyme, very, very important, DNase enzyme. So DNase digest the DNase, the DNase enzyme digest DNA, but RNA proteins are there along with live R strain. So here no transformation principle occur. And a mouse will survive here. But in other two conditions, the mouse will die. It indicates it is DNA, not RNA or protein, that is entered from dead S cells into R cell, yeah, R strain, and the R strain somehow converted into S strain. So that is meaning of transformation. See here. So heat killed S cells, lipids they are removed earlier, carbohydrates also they removed earlier. So remove the lipids and carbohydrates from a solution of heat killed S cells, and proteins, RNA and DNA will remain. Then in first they added proteinase enzyme. Sample should contain here, but no protein because protein are digested. So add then R cells. S cells here appear in a transforming taking place. Add ribonuclease, that is RNase enzyme. Here again you can write the same RNase. Yeah, what you called ribonuclease. Sample should contain no RNA because they will digest again R cells, so they convert it into S cells. So here you can see transformer principle occurs. But when they add deoxyribonuclease subject to solution to treatment of enzyme to destroy either the protein, RNA or DNA. So no DNA. So therefore, when they add the R cells are here not converted into S cell. No S cells appear. Means no transformation occur. So add a small portion of each sample to a culture containing R cell. Observe further transformation has occurred by testing for the presence of virulent R cell. So conclusion, transformation cannot occur unless DNA is present. Therefore, DNA must be hereditary material. So this is the conclusion done by these three scientists from the transforming principle. Okay. Now, so protein of heat killed S strain is not the genetic material from this experiment. RNA of the heat killed S strain is not the genetic material. So the DNA of heat killed S strain is the genetic material because DNA digested with the DNase enzyme when it is mixed with the R strain, unable to transform the R strain to S strain. Okay, that is the conclusion. Now the third important experiment in the search of genetic material with a Hershey Chase experiment. It is also called a blender experiment. Alfred Hershey and Martha Chase. So Hershey and Chase grew some bacteriophage virus. So this is bacteriophage In 11th standard, we have studied the diagram. Bacteriophage are those viruses which infect bacteria. So they contain either that is DNA, genetic material, and protein coat. Okay. So the another characteristics of this bacteriophage, that protein coat is rich in sulfur, not phosphorus, and DNA contain phosphorus but not sulfur. So they grew this bacteriophage virus on a medium containing the radioactive, okay, phosphorus P32. Remember, and some others bacteriophage on medium containing radioactive sulfur, that is S35. Okay, you know radioisotopes 35, 32. So virus grown in P32 medium, they got only the DNA as a radioactive because it is a radioactive isotope. Because only DNA contain phosphorus, okay, no sulfur. A virus grown in the S35 got radioactive protein, that is outermost coat, because protein contains sulfur but no phosphorus. So this is how you can see medium containing bacteriophage having protein labeled with S32. You can see the color, and here blue color indicates medium containing bacteriophage having DNA labeled with P32. Here S35 and here P32. Okay, so these viruses separately infected allow to infect a particular virus, that is E. coli, particular bacteria, E. coli. So these preparation were used separately to infect E. coli bacteria. After infection, okay, so the E. coli cells, that is Escherichia coli cells, were gently agitated. Okay, agitation. ऐसा हिला दिया. They are also called blending. Therefore, it is called blender experiment. You can see here it is a blending. Okay, ये blending आपको नजर आ रहा है. After infection, the E. coli cells were gently agitated in a blender to remove the virus particles from the bacteria which are outside. Jo entered inside, they are there only. Whatever it is attached outside, okay, they are all removed. So then the culture was centrifuge here, see centrifugation, to separate the lighter virus particles from heavier bacterial cells. Okay, so these are three steps: infection, blending, and centrifugation. See, the uppermost part here is called, you can see here, radioactivity, that is phagoprotein in liquid, and here the bacterial pellet, that is higher density, and here the radioactivity phase DNA is pellet here. Okay, so blue color इधर नजर आया और ये जो color दिख रहा है ये इधर नजर आया. Now just to see from your textbook, you draw this diagram. Three steps. Number one, infection. Number two, blending. Number three, centrifugation. Radioactive bacteriophage labeled with radioactive P32, which will label DNA. Radioactive S35 label the only protein capsule, red color. Either you can see. Both bacteriophage allow to infect the bacteria separately. Then after infection, then allow to blending, that is agitation. And after blending in next generation, so they centrifuge, and you can see here result. No radioactive S35 detected inside the cell. So nothing is found here inside the cell. Means DNA not entered, and DNA is entered, but here DNA is not labeled with isotope. So therefore it is not visible. But the radioactivity S35 detected in supernatant, that is upper part. Here radioactive P32 detected in cells because DNA is entered, and here DNA is labeled with P32. Here DNA is not labeled with P32. Protein is labeled with S35. So protein enter नहीं किया. DNA is entered inside the cell. So therefore more radioactivity is found inside the cell, and no radioactivity detected in supernatant. तो सुपरनेंटेंट में जो है इट इज ए प्रोटीन और इनसाइड द सेल है इट इज डीएनए. सो देयरफॉर इन बैक्टीरियोफेज द जेनेटिक मटेरियल इज डीएनए, not protein.
In the
Just to
Hershey
watch this
Chase
video.
experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled.
Now you can see here, okay. So this is how the DNA will enter, okay, and here it is remain outside.
In the Hershey Chase experiment, bacterial viruses called phage In the Hershey Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did
This is what you call blending, okay. Vigorous shaking, yeah, blending,
not interfere
agitation.
with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced
This is P32 labeled DNA and there was S35.
in the infected bacteria.
Again, agitation.
This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
So this is direct proof we got that DNA, but not the protein, that carries the genetic information for a new generation in a bacteriophage virus. Okay, direct evidence is given by this Hershey and Chase experiment. Now just you see experiment, blender experiment, how you can write in the examination. You know those 3 steps, diagram, very important. So they grew some viruses on a medium having the radioactive isotopes, phosphorus, and some other on medium having that is radioactive sulfur. So here it is a S and here it is a P32 and S35. Virus grown in radioactive phosphorus have radioactive DNA but not radioactive protein because the phosphorus present in a DNA but not in protein. Very, very important. Similarly, viruses grown in radioactive sulfur, that is PS35, have radioactive protein but not radioactive DNA because the sulfur is present in protein and not in the DNA. Okay, so that gives a clear differences कि radioactive labeled material एक में protein है एक में DNA, so which one will enter, that is we have to identify and they identify. So second, that is infection. radioactive phages, yeah, bacteriophage virus were allowed to attach, yeah, infect E. coli bacteria. Experimental bacteria is E. coli. So the phages transfer the genetic material to the bacteria, but we don't know which one is entered. Then blending. So the viral coats were separated from the bacteria surface by agitating them in a blender. Yes, sudden shaking. Then centrifugation. So the virus particles were separated from the bacteria by spinning them in a centrifuge machine. So पहिला growing, infection, blending and centrifugation. What is the observation? Bacteria infected with viruses that had radioactive DNA were radioactive and no radioactivity in the supernatant. So bacteria infected with a virus that had radioactive protein, that is S35, were not radioactive but radioactivity found in the supernatant. Okay, so here more radioactivity is found in the supernatant here, and here there is no radioactivity in the supernatant. So conclusion, DNA is the infecting agent that made the bacteria radioactive. Hence, DNA is the genetic material and not the protein. So this is what we got, the direct proof. Okay. So these three experiments to remember. One is Griffith transforming principle. Then biochemical analysis given by Avery, MacLeod, McCarthy. And the third experiment, that is blender experiment given by Hershey and Chase. Very, very important. तीन में से एक एग्जाम में जरूर आता है. Bacteria which was infected with viruses with radioactive DNA were radioactive, indicating that that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with the virus with radioactive proteins were not radioactive. This indicates proteins did not enter the bacteria from the viruses. Okay, itna yaad rakho. So DNA is the genetic material. Okay, now we came to know that why, which is the genetic material, whether it is a DNA or whether it is a RNA or whether it is a protein. Okay, so I have taken this earlier to understand. In your textbook, it is given later on. Now, so to become a genetic material, what property should be there? Properties of genetic material. First property, the genetic material should be able to generate, yeah, multiply its replica. That is called replication. It should be replicated. Second, it should chemically and structurally be stable. Stable hona hai. It should not undergo changes. Third, it should provide the scope for slow changes, what we refer as mutation, and that are required for the evolution. Mutation होने के लिए उधर scope चाहिए. So these three properties are very, very important to become genetic material. Now, where these genetic materials three are applicable, whether they are found in the what we have decided a DNA, okay, इसमें है, yeah, whether it is can found in a RNA or whether it is in protein. But now we have seen DNA, and very important, whatever the changes are there, it should be able to express itself in the form of Mendelian characters, that is dominant, recessive from one generation to another generation. Now, so DNA is a genetic material. We came to know that. Why? Why not RNA? So RNA is unstable because of two reasons. Number one, so there is a 2 prime, okay, at a 2 prime OH group. We'll see that in the nucleotide structure also. Present at every nucleotide, ribose sugar, they have additional OH group in RNA, which is reactive group and makes the RNA liable and easily degradable. So RNA is more reactive than DNA because in DNA, instead of OH group, there is a H group, only hydrogen in second carbon. Second reason is RNA is also now known as catalyst because it is work as a catalyst. Hence, it is a reactive. And most of the earlier reactions are occur around the RNA. RNA is unstable and mutate very, very fast. So therefore, you might have heard the viruses which are having RNA, they undergo mutation. So consequently, the viruses having RNA genome and having shorter lifespan mutate and evolve faster. So therefore, viral diseases are very, very pandemic and they are globally spreading. And within short period, they will also do the changes. Now, so why RNA is unstable? We have seen the two reasons. Now, second, better genetic material, whether it is a DNA or RNA. See, presence of thymine at the place of uracil. Thymine is present in DNA and uracil is present in RNA. So this will confirms more stability to the DNA. Both DNA and RNA are able to mutate. दोनों में भी mutation होता है. But as we have discussed, RNA being unstable because of that OH and highly reactive, it mutate at a faster rate. तो जो third property था दोनों में लागू होता है. RNA can directly code for the synthesis of proteins. Hence, it is easily expressed. The DNA cannot code directly, so it cannot be easily. However, the DNA depends on RNA for protein synthesis. So again here misnomer. Then who came first? DNA पहला आया क्या RNA पहला आया? Which is the first genetic material. If you see the property of RNA, it is catalyst. Okay, in all in chemical reactions, in all in protein synthesis, so RNA is the best first genetic material. But here, which is the most stable genetic material? DNA is the genetic material. The protein synthesis machinery has evolved around the RNA. Okay, so RNA is first genetic material, but the better genetic material is DNA. Now see here, we have just list out. Reasons for stability, less reactivity of DNA. Reasons for mutability, that is high reactivity of RNA. First reason, DNA is double stranded. You can see here, a DNA is double stranded. But RNA, you can see here, single stranded, a single stranded. So in DNA, so there is a presence of thymine here. You can see thymine. This is the thymine, nitrogen base. But here in RNA, so there is a presence of uracil. Here uracil, that is U. And the third reason is absence of, that is 2' OH in sugar. You can see here, this is 2-deoxyribose. So here only H is present. Remaining OH is there. But at second carbon, only H. But here presence of that extra OH in 2' ribose sugar. So these three, double stranded, presence of thymine, absence of additional OH group makes the DNA more stable. Single stranded, presence of uracil, presence of extra OH group at 2' make the RNA highly reactive and more unstable. So therefore, DNA is the best genetic material, but as I said, all the biochemical reactions occur around the RNA. RNA is the first genetic material. So some of the RNA, they somehow modified into DNA. Yes. Now, better genetic material, again, that is for the storage of a genetic information, we have seen for the storage of, okay, a genetic material. Here we can conclude for the storage of genetic information, DNA is better due to its stability, but for the transmission of genetic information, RNA is better because it will directly code for the proteins. RNA can directly code for the protein synthesis, hence can easily express the characters. So therefore, DNA is dependent on RNA for protein synthesis. And this is what we call central dogma. So there is a DNA. DNA store the information in the form of code, yeah, genes. So these genes, yeah, codon is transcribed in the form of RNA transcription. From this RNA, so protein synthesis takes place. So that is called translation. Okay. So this is how you can conclude that DNA is the better genetic material compared to RNA, but RNA is the first genetic material. So thank you for watching the first session, and you keep watching my second session with of this chapter. Thank you one and all, and keep subscribing the Gopal Biology channel. Thank you.